Hello, everyone. This is the lab makeup narration for exercise 4 and 5. This is the September 4th lab that we couldn't hold because of the Labor Day Saturday. So what you'll see here in this recording is models. Many of the aspects of the lab will be coming to you almost like if you would be in the lab, but because of the inability to carry out a conversation, some parts of this labs I'll just mention, but we can't really spend time on it. And I mentioned these as we go along. So when you are listening to this narration, it's good to have the lab report on exercises 4 and 5 next to you. And you can take notes of it. And when you come to the lab on September 11th, please ask any questions. I'll be willing to stay in the lab after our regular time and bring everybody up to speed on, for example, microscope slides or any other questions you might have. So the lab report is attached next to this narration, and it's a PDF file. It will probably have the title of Lab Report on Exercise 4 and 5. So after this introduction, let's have just a little general intro to the exercise 4 and 5. We are in the domain of the cell, and during this week, you are learning about cells for the lecture portion of this class as well. In the lab, we take a much simplified view, focusing on available models, microscope slides, and at the very end of this lab, you'll see an experiment that's also, again, reflecting how we approach this topic in the lab. So what you see here on the screen is our wonderful model of a cell. And what's most relevant to the description of all these labeled parts is table 4.1 in your lab manual. It's on page 35 in my lab manual. The first line of the lab report is just stating, we reviewed the organelles and their functions from page. So write down the page number, so you will be reminded where this table is in your page. Very useful table. So in this table, you see 3 bolded lines. Those are the regions of the cell. And the first region is the plasma membrane. So when you look at this thin shell, that represents the plasma membrane. And so the plasma membrane functions mostly as a barrier, kind of saving or protecting the internal structures of the cell, and also preventing potentially harmful influences from the outside, whether it's mechanical, but mostly chemical influences. So the plasma membrane is a barrier. In exercise 5, we will see how in some instances this barrier can be penetrated by molecules that are important to bring into or move out of the cell. A lot more details coming about the plasma membrane in exercise 5. So your lab manual mentions microvilli. Please make a note of it that microvilli are not visible on this model. In your chapter 3 lecture material, there's a lot more mentioned about microvilli. We will be seeing that the role of microvilli in AMP2 mostly. Microvilli are relatively short extensions of the plasma membrane in general to increase surface area of the cell. The second large region of the cell is the nuclear region. You see 2 parts here. And the second item in your lab report is asking you to compare the function and the composition of nucleus to that of the nucleolus. So nucleus is much larger, spherical component. Spherical structure. The major biological component of the nucleus is DNA, deoxyribonucleic acid, and the function of the nucleus is to store hereditary information. Our genes are located on those DNA molecules, the chromosomes inside the nucleus. In a smaller corner of the nuclear region, there's something totally different. This is the nucleolus. The chemical makeup of the nucleolus is mostly RNA, ribonucleic acid, and the nucleolus has also a totally different function. It is making ribosomes, so the nucleolus is a factory, and a nucleus, you can look at it like a library. In between the plasma membrane and the nuclear region, there's this ocean of organelles. Your lab manual mentions it. I don't think it's labeled on this model. There's this shiny, translucent part of this model. That is the cytosol. It's a sticky solution that surrounds all the organelles and internal structures of the cell. We will play a lot more attention on the organelles and internal structures. So let's review these in the order of the lab manual. The first organelle here is mitochondria. You can see these orange ovals. There are other places in addition to the labeled ones. Mitochondria function as the organelle that makes ATP. You have seen the role of ATP in lecture material a number of times. So this is the linkage to the actual location where it is being made. Second organelle is the ribosome. Ribosomes are showing up as white dots, and you can see dozens of them. Look all over the model. Ribosomes function is the site of protein synthesis. So ribosomes make protein. Right off the bat, we can see some organelles that are lined on the outside, surrounded by ribosomes. There's rough endoplasmic reticulum. The rough endoplasmic reticulum has a role in processing, transport, modification of new proteins. Rough endoplasmic reticulum is also the location for phospholipid synthesis. And from lecture material, you'll see a good deal of explanation how phospholipid makes up most of the plasma membrane. So it's an important factory located in the rough endoplasmic reticulum. We also have a smooth endoplasmic reticulum. There are no white dots around this organelle. Smooth endoplasmic reticulum functions as a site of steroid synthesis. The smooth endoplasmic reticulum also have enzymes that function in detoxification chemical reaction. The next organelle in this table is the Golgi complex. Sometimes it's called Golgi body, Golgi apparatus, all those names are fine. The Golgi looks like this pink stacked plastic saucers or saucer-shaped structures. The Golgi is transporting, modifying, and sorting proteins. So those proteins are gaining their final working shape in the Golgi, and then the Golgi sorts the new proteins. The Golgi dispatches some proteins to the plasma membrane, some to the lysosome, some to rough endoplasmic reticulum. So it's Golgi who sends the right proteins to the right location. We also have secretory vesicle in this model. This is not labeled. You might want to make a note of it. Right here is the best version of secretory vesicle. When proteins need to be moved or other material need to be moved to the outside of the cell, secretory vesicles merge with the plasma membrane and they shove the content to the outside. If this cell is a beta cell in our pancreas, this cell would be making insulin hormone, and insulin hormone would be pushed to the outside of the beta cells so the hormone can get into the bloodstream and fulfill its function as a hormone. We'll be seeing secretory vesicles in the muscle and nervous system chapters, which are chapter 10 and 12, in a little different context. We do have another organelle, lysosome, labeled. Lysosomes are small bag-like structures that are loaded with digestive enzymes. The enzymes in the lysosome can digest carbohydrates, proteins, lipids, DNA, and some other material as well. Lysosomes function in degrading or removing any organelles or part of the organelles that could be damaged. Lysosomes can remove surplus organelles from the cell, and especially in white blood cells, lysosomes can digest material that was engulfed and brought into the cell as well. In chapter 3, you will see the function of lysosome in digesting material that's coming into the cell from the outside. You'll see several examples of that. Make a note of it that the next two organelles, peroxisomes and cytoskeleton, are not on this model. You'll see a lot more elaboration, especially on the cytoskeleton in lecture material. Centrosomes, located right here. So here's a centrosome with two centrioles. Centrosomes will come our way towards the end of this exercise 4 when we talk about cell division. The centrosomes function as the origin of spindle fibers that are used to move chromosomes during cell division. But centrioles are also the core movable part of cilia and flagella. So the centrosome and the centrioles have a variety of functions. When it comes to cilia and flagella, you'll see a lot more exploration in the lecture material. And those two organelles, cilia and flagella, are the last one in the table. We don't see them on the cell model. So in the lab, you're not responsible finding them on this model. In your lab report, points 3, 4, and 5 are examples to illustrate the dynamic nature of the organelles. When you look at this plastic model, nothing could be further from the truth. When you think about living cells, cells are actively working, living part of our body, and those cells need to change, adapt to the environment, follow instructions. So those examples 3, 4, and 5 are for you to consider at home. Again, in the lab, we'll be having more of a student-led discussion about those points 3, 4, and 5 in the lab report. Number 6 in the lab report is stages of mitosis. So the cell, when it comes to creating new cells, has this process at its disposal, mitosis, the division of the nucleus, and then division of the rest of the cell. So cell division under the name mitosis is happening in cells in areas of the body where we have to replace old or dead cells. We have constant cell division in our small and large intestine to replace the lost cells that are on the inner lining, which are dragged away and digested daily. Our entire intestinal cell lining is replaced every 5 to 7 days. So those cells in our intestines are eminently able to carry out cell division in producing 2 cells out of this single one. We can also list a good example for cell division of the embryonic and fetal development. We all start out from a single cell to make several trillions of cells that make up the adult body. Not only we have to create new cells during embryonic and fetal development, during our growth after our birth, also is the result of cell division. So you see here an illustration from the lab manual. I believe your textbook has this too. And so cell division comes in with phases. What you see here, the entire circle, is called the cell cycle. Before we go any further, it's important to say a few words about mitosis and the other cell division called meiosis. So mitosis, as you see here, the individual steps that carry out this cell division. Mitosis has a starting cell, which is right here in the interphase. The starting cell has 46 chromosomes. This is the parent cell. And the daughter cells, as you see here, these 2 daughter cells also will have 46 chromosomes in human beings at the end of mitosis. When you say meiosis, M-E-I-O-S-I-S, meiosis. Meiosis has the parent cell starting with 46 chromosomes, but the daughter cells will only have 23 chromosomes, half the normal human chromosome number. In meiosis, the cells, the daughter cells are reproductive cells, either sperm or oocyte. So meiosis is found only in 2 organs, ovary in females and testes in males. Mitosis is found in virtually every tissue where cell division is happening. So mitosis is the most frequent and day-to-day employed cell division. We're going to say a few words about these phases in connection with the microscopic appearance of the cell. So let's start with a cell that is preparing for cell division. The cell that you see here is in interphase. Now, interphase cell has to duplicate the DNA, has to duplicate cellular organelles. So while it doesn't look like a lot is happening in this cell is crazy busy creating new organelles and doubling the chromosome number. Typical appearance under the microscope is that you can see the nuclear membrane, and inside the nucleus there's this fuzzy pinkish material. So for this portion of the lab, it will be essential to check out the microscope slides that we do have in the lab. They look a good deal different. Magnification will not even be close to this, so it'll be good to train your eyes on that, on those slides. And so if you come on September 11th, Saturday, after the lab, you are welcome to check these cell division stages. We'll have microscope set up for your practice. So in the cell cycle, once the cell division mitosis actually begins, we see prophase. Up here, you also see another prophase. Take a look at the nuclear membrane disappeared. What we have here is the chromosomes becoming like stick-like structures. You can see it in both cases. And so this is the prophase. Mitosis, the division of the nucleus, has begun. In metaphase, the chromosomes align in the middle. Now you can see the centrioles, the spindle fibers that grew out of the centrioles. In anaphase, along those spindle fibers, the chromosomes are now moving to the opposite pole of the cell. So we are separating two sets of chromosomes, one set for each daughter cell. And you can see the centriole and spindle fiber yet again. I'm going to digress a little bit. Take a look at up here to the left. This is in interphase. Right? Okay, moving on. So the last phase of cell division is called telophase. In telophase, not only that the groups of chromosomes for each cell separated, but the daughter cells are now forming new cell membrane to eventually totally separate from each other. The separation has not occurred, but it's very close. This is telophase. This big gap, indentation, where the two cells are preparing to separate, this is called a cleavage furrow. This is a typical structure to recognize telophase by. And before I switch away from these microscope slides, it's important to mention just what exactly did you see and will you see under the microscope when you take a look at these on September 11 on Saturday. So the cells that you see here are whitefish cells, and this entire set of hundreds of cells represent a fish embryo. So the model for mitosis that you have seen is called a whitefish blastula. Cell division occurs at a breakneck pace, and it's a real great model. Chromosomes are staining dark blue or black, and so it's really easy to see. So this is why we use a whitefish and not human cells for the visibility and the clarity. Number 7 in your lab report is asking you to consider a breast cancer patient. Think about this at home, ponder what phases of mitosis would you see in the cancer of a breast cancer patient after paclitaxel treatment. That's best thing to consider for home in this narrated version of the lab. So with the mitosis, we have completed exercise 4, and this drawing that you see here, I usually put on the board line by line. This is part of exercise 5. Exercise 5 in this lab is material about transport across plasma membrane. So on this ingenious drawing, here's the plasma membrane. See cell membrane. And you see arrows pointing across the cell membrane. And you can ask yourself, didn't we just say on the cell model that the plasma membrane is a very effective barrier? So let's say a few words, a few scenarios when this barrier must be broken, it must be overcome. So let's start up here inside the cell. Imagine that a living cell, we just mentioned the organ of mitochondria, they make ATP. The cell must have ATP, so mitochondria are churning out, manufacturing ATP 24/7. In that process, mitochondria use oxygen So mitochondria effectively vacuum oxygen outside from inside the cell. So oxygen concentration inside the cell will be low. At the same time, mitochondria spew out a large amount of this waste product, carbon dioxide. Carbon dioxide concentration inside a living cell are high because this waste product keeps building up. So if the plasma membrane is not going to cooperate, then this cell is going to die very shortly of oxygen deficiency and the buildup of this waste product. Outside the cell, oxygen concentration is high because mitochondria are not using it, and carbon dioxide concentration is low because mitochondria are not making any outside the cell. You see the arrows. This is the desired movement of these gases. And when you look at a living cell, indeed, oxygen is constantly streaming into the cell. Carbon dioxide is constantly streaming out of the cell. So two important aspects here. The plasma membrane is not a barrier for gases, so they can move any particular direction. But then let's ask ourselves, why oxygen is moving in one direction, carbon dioxide is moving also in one direction? And the reason for that is that gases and every molecule in this universe always moves from a place of high to a place of low concentration. This is the preferred way thermodynamically for these molecules. In plain English, you can think about it that, well, oxygen at high concentration needs more elbow room and it wants to move to a place where there's more spacious, low concentration. Any way you put it, this is a common natural process. The name of movement of material from a place of high to a place of low concentration is called diffusion. Carbon dioxide also diffuse from the inside the cell to the outside, following its concentration gradient, moving from a place of high to a place of low concentration. And so the cells are happy because their oxygen supply is provided courtesy of diffusion, and carbon dioxide will be leaving the cells yet again courtesy of diffusion and the concentration difference on the two sides of the plasma membrane. Mitochondria are also using glucose as a starting material. And so glucose needs to be streaming into the cell to supply ATP production. Inside the cell, the glucose concentration is low, or almost zero. Outside, glucose concentration is high because from the blood sugar glucose is keeping this concentration high. So you might think that, okay, well, the glucose is just going to diffuse into the cell from a place of high to a place of low concentration. And here comes the difficulty. The plasma barrier is a really, plasma membrane is a real formidable barrier for glucose. So glucose on its own runs into the plasma membrane and bounces right back. It cannot go through. So glucose needs help to follow its movement by diffusion. From the cell, a transporter protein is made, and this transporter protein grabs onto a glucose molecule at a place of high concentration and flips it over across the membrane into the place of low concentration. Because the movement of glucose is still moving from a place of high to a place of low concentration, it is considered diffusion, but it is now helped out, facilitated by the transport protein. So glucose is moving through facilitated diffusion into the cell. Excuse me. Another way molecules move across the plasma membrane is through channels. We use water as an example because water movement is described by a totally different term. It's called osmosis. Water always moves from a place of low salt to a place of high salt or high solute. So the channel for water molecules is called aquaporin. And you can see in this case, water moving into the cell. We're going to have a really good illustration of this using a red blood cell model from your lab manual. So let's take diffusion, facilitated diffusion and osmosis. And we can put these three transport processes together into a category of passive transport. Passive transports are the movement of molecules across plasma membrane that does not need ATP, does not need cellular energy. This is relatively cheap for the cell, and our cells rely on for the movement of a number of components across the plasma membrane on passive transport. Here in the corner is a topic that will be hugely expanded on in your lecture material, active transport. Active transport use ATP as opposed to the passive transports that don't. And the active transport is performed by pumps. So you'll see pumps in chapter 3 material. So these pumps and active transport, again, will come our way in the lecture material in much, much more detail. Here in the lab, we don't really have a good way to demonstrate it, as opposed to the passive transport, but some of that you'll see demonstrated. So these were the examples that we considered just to take a look at the basic transport processes across the plasma membrane. This illustration is from your lab manual, and let me find the page number. This is page 48 in my lab manual, and I'm looking at figure 5.3. So here you see a red blood cell, which is typically donut shaped. So what's the big deal about this panel A? In panel A, the red blood cell is in a salt solution that does not attract water. And so water movement into and outside the red blood cell going at the same speed. The solution outside the red blood cell is called isotonic. Iso meaning the same concentration salt as inside the red blood cell. Water has no incentive to move much to the outside or to the inside, and so the red blood cell maintains its shape in isotonic solution. When you look at panel B, you see something totally different. The red blood cell inflated like a balloon, and you see all these gray arrows pointing into the red blood cell. These gray arrows represent the movement of water molecules. Now, why would water molecules move into the red blood cell? That is because we changed the solution. So in panel B, the solution is hypotonic, low salt. When the solution outside the red blood cell has lower salt than inside the red blood cell, water molecules always move to a place of high salt or high solute. In this case, inside the red blood cell. So in hypotonic environment, hypotonic solution, red blood cells will inflate, and sometimes the event hemolysis occurs because the red blood cell membrane bursts and hemoglobin is just spewed out into the environment. So panel B is the look of a red blood cell in hypotonic solution. In panel C, we see something yet again different. The red blood cell collapsed, shriveled, and you see these gray arrows pointing from the red blood cell into the environment. Again, gray arrows represent movement of water, osmosis. Now, why would water move out of a red blood cell? Again, we changed the solution on the outside. And now on the outside, we have a high salt, hypertonic solution. When water molecules inside the red blood cell see that on the outside there's a high salt, hypertonic solution, water molecules again are attracted to high salt, high solute areas, and water molecules then move into the surrounding solution by osmosis, and the red blood cell shrivels. This shriveling also has a special term to it. This is called carenation. C-R-E-N-A-T-I-O-N, carenation. In your lab report, this table is point 12. 9, 10, and 11 again are public discussion areas that we did in the lab. Please take a look at these and see what you think on your own at home. If you have any questions, you can always message me, or we can talk about it on Saturday after the regular lab. So going to the last page of your lab report, point 13, here, what we did in the live lab is an actual experiment. I demonstrated it. So here is the experimental setup. We have a big container of tap water. We have, as you see it on the balance here, an artificial cell. Into a dialysis tube, I placed a red colored solution. I weighed the bag, and then I placed this bag inside this container of water. Our hope is that we can demonstrate diffusion and osmosis in this experiment. So it takes about half hour for this experiment to run. So let's see, why would we expect the demonstration of osmosis and diffusion? Here is a little detail of what's inside and outside of this dialysis bag. So here's the artificial cell wall. It has pores, and molecules can move through it following their concentration gradient directions. Take a look at the inside. There is a red dye in high concentration. There is sugar, regular cane sugar in very high concentrations. And of course, there are water molecules, this blue triangle. On the outside of the dialysis bag, in this container of water, there's nothing else but water. So this is all water molecules on the outside. So how could we possibly visually demonstrate demonstrate diffusion? Well, we can follow the color of the red dye, and because inside the bag, red dye has high concentration, on the outside the bag, the red dye has low concentration, diffusion should take the red dye to the outside. We could visually detect the result of diffusion, the movement of red dye from inside to the outside of the bag, by looking at the color of the water in the bag. I'm sorry, the water in the container. So let's take a look. Here is the color of water. A lot of red dye indeed entered the container. So appearance of color in the water outside the bag is our evidence of diffusion. So in your lab report, A is demonstration of diffusion by observing red color in the water outside the bag. B in your lab report is demonstration of osmosis. So as this bag was sitting in this water-filled container, the following occurred. I'm going to go back to the previous image here. So what is the natural tendency of water molecules to move from a place of low solute, such as outside, to a place of a high solute. Inside this bag, there's a lot of sugar, red dye. So water molecules see this environment as high solute, and they move in to the bag from the outside. Now, we cannot visually observe the water molecules inside the bag, but because water molecules are trapped inside the bag, we can weigh the bag and ask the question, was there any water trapped inside the bag? Did the weight of the bag increase at the end of this experiment? And you have to take my word for it. Those of you who observe this in live, they can attest to that indeed the weight of the dialysis bag increased. And so for point B in your lab report, the last item is we demonstrated osmosis because water entered the dialysis bag, and we could demonstrate increased weight of the dialysis bag. So that is our way of confirming that osmosis indeed occurred as we would expect it. So this is the end of this narration of the lab. I'm going to attach a little note and also the PDF file of the lab report. And if you have any questions, please don't hesitate to ask. I think I'm going to stop right here.